Hey everyone, quick announcement for this episode. The software we use to record remote episodes presented an issue for me this week. It recorded about the first couple of minutes, and that was like the warm up talk, so I don't think that's in the episode, but didn't record the rest, although it pretended like it was the entire time. So when I downloaded my file, I got a 2 minute and 17-second file, while everyone else got their 2 hour and change file. What this means is that none of my audio will be in the episode, which is going to result in a unique listening experience. I talk a bit less during the actual content portion of the episode with Richard. I think it's going to still be a little challenging listening to him respond to questions that I asked, but there's really nothing I could do. One alternative would have been to listen to it, try to remember what I said, and record that and paste it in. But that would lead to a bunch of having to resync after every time that I did that. Plus, I think it would still be pretty disjointed, and it would have taken forever. So we actually recorded this the weekend before Christmas, and I've been reluctantly sitting on it while I attempted to hear back from their support team, Zencaster, that is the service we use, which is overall great and affordable. It's just I think it's my computer or something. Something weird happened. It also happened during the episode that we did with Eliezer Yudkowsky for We Want More, the finale episode. But fortunately, I had a backup recording running during that one because I didn't want to get screwed again. So, anyway, sorry for the sort of jumbled mess you can expect to get during this one. Like I said, there's really nothing that could be easily done about it. I do need to give a big shout out to John Peterson, our patron for this week, since I was the one who named him during the thank a patron section at the end. So thanks, John, you rock, and you'll be able to hear Inyash and Jace agree, though you won't. It'll again sound a little disjointed. So sorry about this, everyone, and especially sorry to Richard for having your first episode on be kind of a wreck, but you came through great. So when you come back on for future episodes, we'll make sure that nothing like this happens again. And thanks for putting up with us, everybody.
Welcome to the Bayesian Conspiracy. I'm Inyash Brodsky.
Richard Acton.
Richard Acton, who are you and why are you on this podcast?
So I am currently a postdoctoral researcher and bioinformatician on the technical staff at a research institute called CCAD in Cologne in Germany, and I study aging biology.
Nice. So the reason you came was specifically because you heard our longevity episode a couple episodes back, and you said you had things to say about that.
Yes, yeah. I heard you were. Yes.
Yeah.
And as a long-term fan of the show and some of your other works, I thought I'd take the opportunity to contribute.
That is awesome. That is I was going to say that's the best part of having an audience of people who are really into tech and research stuff, that every now and then someone is like, I know something about this, let me help you guys out. And that's awesome because we learn a lot more, and hopefully our audience does as well.
Yep. And scientists are always looking for interesting outlets to share their work, right? Because most people we spend most of our time talking to know about what we do because they're in our niche, so they're probably a little bit bored of it. You talk to other people, then
did you say
yeah.
Oh, go on.
Oh no, go ahead.
Did you say biomathematician?
No, bioinformatician. I am not a mathematician.
Well, I mean, that's just as confusing. What is a bioinformatician?
Okay, yeah. So I mean, a little bit more clarity on what I actually do day to day. So I have a weird split because I'm 50-50 between a technical role and a research role. So I'm in the lab of Bjorn Schumacher, where I do research work, analyzing data for my own research questions and for others in that group. And then in the technical role, I'm doing a bunch of research analysis work, a lot of sequencing data analysis for other people in the institute, and setting up some of the core bioinformatics infrastructure. So at the moment, I'm trying to learn a bunch of infrastructure as code stuff for Docker, so I can set up a server for running a bunch of bioinformatics tools. But yeah.
What would those tools do?
The one I'm trying to work on is basically to set up a tool to let other researchers with less command line experience use a bunch of bioinformatics tools that otherwise are very difficult to use. It's called Galaxy. But yeah, we're getting a little into the weeds there now.
So what is the bioinformatics thing? Is that information on biological systems?
It's data science for biology, basically.
Okay.
Yeah, and well, I mean, to be honest, like the bioinformaticians came first, and the data scientists kind of, in many ways, branched out from a lot of stuff that was initially in bioinformatics, because genomic information was kind of some of the first data sets that were really big. So the sort of the big data bioinformatician, the big data scientists that we know today, a lot of them had kind of a start as sequencing data really started to get big.
So you guys are like the data hipsters, and all these people are like just new on the scene, trying to pretend like they were here.
We're all writing in Perl, and yeah, well, we moved on, but still.
Cool.
Yeah, I mean, I can offer the perspective of someone working in the field. So I'm not I'm not super current specifically on the sort of like longevity interventions that, I mean, I could talk a little bit to those, but like that's not really my area of expertise. I'm kind of more about the basic biology underpinning aging, and specifically the epigenetics of aging. Yeah. Okay. Mm-hmm. Yes. Would you like me to give the spiel on that again? Okay. Sure. So I'm co-hosting a podcast with my friend Michael Glinker, where we are talking about a sci-fi series of books by Octavia Butler. It's the Xenogenesis trilogy. We're talking about the first one, and it's kind of in the style of We Want More or We've Got Worm, where I'm Stephen and and Michael's Brian. Yeah. So I've read it, and he hasn't, and I get to have fun about that.
Awesome. And what is the podcast called?
It's called Xenothesis. You can find it at xenothesis.com.
Awesome. I have not.
Yeah, Xenogenesis is the trilogy, also for some reason rebranded as Lilith's Brood later after the fact in like a re-release, and then
I didn't realize those were the
Dawn
same thing. Okay. Yeah, I've read Wild Seed, and like I have of that book and a few of her short stories that I've read, I've loved them. I just there's so much out there to read. I don't, I'm
there's
forever
only
catching
so
up.
much time. Yeah.
But
the great thing about this series of books was we're both biologists, me and my co-host, and there's a bunch of really interesting biology that gets kind of a much more, I think, fuller treatment in this particular sci-fi story than it does in many other sci-fi stories. That's why I picked it.
How far through the series are you so far?
I just edited our 19th episode.
And how many do you think they're going to be total?
I forgot to compute that. I should have checked at some point.
How far into the trilogy are you at this point then, I guess?
So we're only in the first book. I think we're probably about 2/3 of the way into the first book now.
Okay, so probably about 90 episodes altogether.
Yeah. If we decide to carry on directly into the sequels, we might do something else. We're not entirely certain yet. Yeah.
Awesome. That is really exciting. I love that there's more and more of these because, like, it's just more fun reading books when you have these sorts of analyses.
Yeah. It's I loved reading Worm with We've Got Worm as a companion. It was
I have a book club where every 2 weeks we get together and meet and talk about the book that we all read together over the last 2 weeks, and that really makes reading a lot more fun to have someone else to talk about it with later.
Absolutely, yeah. It's a great excuse to hang out with friends podcasting as well, right?
Yeah, absolutely.
You have an external commitment to keep to the schedule.
Which as the older you get, the more you need that.
Yeah.
You no longer feel like it's okay just to like go over to someone's house and hang out for a few hours.
Yeah, everything's got to be on the clock for something or other.
Like I could hang out, or I could use those few hours to get this done. There's always more to get done.
Always.
So, before we get into the meat of the episode, shall we do our Less Wrong post like we normally do?
I'm here.
Hey, just in time.
Up by being like in the middle of somebody talking.
Yeah, I'm currently visiting my parents for Christmas, so I'm in Farnham in the UK. But yeah, I moved to Germany 6 months ago, so I'm normally in Cologne.
Huh.
It's awesome.
Yeah.
How many time zones different is Germany from Britain?
Just the one.
Just the one. Okay. I realized as I was asking that, I'm like, I could just Google that. I don't know why I'm wasting the time of someone with expertise in bioinformatics on stupid time zone questions.
Well, evidently it needs to be done more because that way I would get the math right when I do it.
Well, yeah. I was able
Oh,
to
yeah.
go out and get my morning walk, so it worked out fine.
Nice.
Yeah. All right. So as I was saying, the less wrong posts. Shall we jump into those?
Absolutely.
All right. The first one actually in sequence is Petrov Day, which we are not going to get into because we've spoken of Petrov Day several times before, I believe. I think the one that we had the most to say on it was episode 69, Culture War 2.0, which was 2 years ago on Petrov Day that came out, or close to Petrov Day, when someone pushed the button on accident during one of the Petrov simulators, which we had to comment on because it was pretty awesome. Yeah.
We had a really good Petrov Day little rationalist meetup here.
Yeah,
We did the ceremony with the candles.
Nice.
You know what? Next year, maybe we should have the ceremony and then report back on it afterwards for the episode. Because this year, a bunch of us just didn't go due to a combination of COVID and the fires that made the air hard to breathe.
Yeah.
Yeah.
The pandemic and and everything was on fire.
Yeah.
It's a weird time to live.
So Petrov Day came up in our podcast too. Actually, it's because it's a post-nuclear war world. It seemed relevant.
Yeah. I love the fact that we've sort of got our own holiday thing going.
Yeah, nice to have some rationalist rituals, I suppose you might call them.
But
yeah, it takes a while.
What we really need to do is be born into rationalism and then have our parents raising us with these rituals as if they're a normal thing. Well, no, I mean, it should totally be opt-in, but Christmas doesn't feel weird because we've all been surrounded by Christmas since we were born, right?
Yeah, opt-in gets complicated for children.
Yeah. If I were to move to a new society where it had something like Christmas, but I'd never been around for it, I might think it was strange.
I still think it's strange. I mean, I went to Walmart the other day and there was just everything garishly lit and horrible Christmas music playing and all the employees wearing
you mean wonderful Christmas music? Oh, I love
so
Christmas.
aversive.
At least that's actually in December, right? If it's before this month, then it's...
Oh yeah.
Oh, in America,
it hasn't even been
November
Thanksgiving,
as well.
and there's Christmas stuff.
Yeah.
I feel like it's Halloween and then Christmas now immediately.
Yeah, Thanksgiving has been completely absorbed by the Christmas monster. Oh my god. I know.
Maybe they were celebrating to have Petrov Day. That's the 26th.
The 26th, yep.
Yeah.
All right. So.
Okay, well, I think Eliezer put it best, so I'm just going to read his post, How to Convince Me That 2 + 2 = 3. He starts it he starts it off by saying that rationalists put a heavy premium on the paradoxical seeming claim that a belief is only really worthwhile if you could, in principle, be persuaded otherwise. Saying if your retina ended up in the same state regardless of what light entered it, you would be blind. But then a poster commented that I cannot conceive of a situation that would make 2 + 2 = 4 false. And so Eliezer wrote this post in reply, which I just wanted to comment on real quick. This is the coolest thing that it wasn't just a series of missives sent from on high. This was a whole community thing with comments going back and forth and people talking, and this post in the sequences is a direct result of somebody commenting on something he said, which is really cool. Yeah. Cool. Yeah, I don't know. I think that made a big difference in what Less Wrong was and, why Less Wrong 2.0 is working as well. Because, again, it's more of a collaborative thing with a lot of people working together than just here's my blog about what I want to talk about.
Yeah, I realized I had this thought, I wish I'd been around when they were being released and when everybody was interacting, and I realized actually I kind of was for Slate Star Codex, which had a really similar thing going on.
Yeah.
Oh,
I'm
nice.
so excited
Oh, yes.
to have Eliezer and Brian interact. It's gonna be so hilarious.
Yeah.
Hmm.
Yeah. But he does have some questions lined up, eh? Okay, so I guess what he draws a comparison to is why you think that 2 + 2 = 4 nowadays. And that's mainly because when you have two things and then you have two more things, you have a number of things that is, 4,
Equal to
basically.
4.
Yeah, it's just the way physical reality is. But he says, suppose I got up one morning and took out 2 earplugs and set them down next to 2 other earplugs on my night table and noticed that there are now 3 earplugs without any earplugs having appeared or disappeared. Moreover, when I visualize the process in my own mind, it seems that making 2 x's and 2 x's come out to 4 x's required an extra x to appear from nowhere. Also, I would check a pocket calculator, Google, and perhaps my copy of 1984, where Winston writes that freedom is the freedom to say 2 + 2 = 3. And none of these were put like directly after each other like that. I'm cutting out a bunch of things in the middle for speed. But what it comes down to is saying that what would convince me that 2 + 2 = 3, in other words, is exactly the same kind of evidence that currently convinces me that 2 + 2 = 4. The evidential crossfire is physical observation, mental visualization, and social agreement. And yeah, he also talks about then he would have to start wondering, why did he ever think 2 + 2 = 4? Was he seen the simulation as someone tried put brain damage on him? What was it that was so wrong about his viewpoint? But I mean, on the one hand, I have an incredibly hard time visualizing how you could have two things, and yet somehow when those are thought of as together, you have 3 things. But if that's the way reality worked, you can't really argue with reality, right?
There's plenty of things that reality does.
Yeah. Well, you couldn't take 2 barrels away again, because then you would have 3 barrels again. Because if 2 + 2 = 3, then 3 - 2 = 2 again.
Yeah, I mean, I think the point that he's trying to make is less about the specifics of arithmetic and more about radical empiricism, right?
I love that the first instinct was to start munchkinning it and start lawyering the munchkinning. Most rationalist response.
Yes.
Well, yeah, it'd be lopsided with only 3 wheels, but you would have 4 wheels, so it'd be okay.
All right, let's figure out how we can game the stock market.
Well, why did you only buy 3 tires, sir?
Maybe all cars would just be those 3-wheeled cars.
I also
Trikes.
It's all a scheme to get you to buy more tires than you need. Yeah. I was reminded, before we move on, of a story that appeared in Strange Horizons, which is a cool online magazine that actually pays professional rates for those of us who try to sell things sometimes. But back in 2000, there was a story called The Secret Number, which I don't remember too much of, except it was a detective murder mystery short story where there's a mathematician that's been killed at the beginning, and his assistant keeps claiming that it was the secret number that killed him because it doesn't want to be found out. And it turns out there's a secret number between 3 and 4 called Bleem. And and obviously the guy doesn't believe him. There's some questioning, how could this work? Blah, blah, blah. He's like, it just does. They're hiding. And so they arrest him. And the next morning, the assistant is found dead. And he's like, oh, it must be a suicide. He felt so guilty for killing the mathematician guy. But then at the very end, there's 3 jelly beans left on the murder room nightstand. And one of the detective assistant picks it up and is like, hey, look, free jelly beans. And the first detective is like, oh, no, don't! And he eats the jelly bean, and he looks down on the night table, and there's 3 jelly beans left. Because the mathematician managed to isolate Bleem jelly beans rather than 3 or 4. Yeah, it's a fun little story.
It reminds me a bit of this really weird movie about the whole P = NP thing, and some mathematician discovers some hacky shortcut to prove that it's the case, and that you can solve stuff in polynomial time, and then there's a whole conspiracy. I can't remember the name of this film, but
are you thinking of Pi?
I don't think so. Maybe.
At the very end?
It's been so long since I've seen it, I can't remember.
Oh, and there's like a cabal of Orthodox Jews that are after him, in addition to like stockbrokers and like all these various powerful groups around the world.
That does sound kind of
I need to know
familiar.
what this movie
Yeah,
is.
because the number that he discovered that solves the PNP problem is also the secret name of God that they've been trying to find forever.
Yeah.
Yeah, it sounds ridiculous, but it's really trippy and cool. It's by the same guy who did Requiem for a Dream and some other like arty movies.
It's like the long version of that chapter in Methods where Harry does the thing with the time turner to do prime factorization with Shor's algorithm.
Oh yeah. Yeah, it was a really trippy movie. And I guess I just gave away the ending, so I'm sorry about the spoilers. Maybe we should beep that part out. Okay, thank you. Yeah, the post ends with, there's really only two possibilities for a belief of fact. Either the belief got there via a mind-reality entangling process or not. And yeah, that's what he was saying. That is how you would entangle your mind with reality, by putting 2 and 2 jelly beans together and somehow getting Bleem jelly beans rather than 4. Yeah. Well, not everything has to have a beautiful segue, right? Sometimes we can just be like, and now let's move on to the next post.
It's so enjoyable watching him create them.
That's true.
Keep it up. All right. There's 2 sealed boxes for auction, A and B. Only one contains a valuable diamond, and there are all manner of signs and portents indicating whether a box contains a diamond, but I have no sign which I know to be perfectly reliable. There's a blue stamp on one box, and etc. There's some things. Now suppose there's a clever arguer holding a sheet of paper, and they say to the owners of box A and B, bid for my services, and whoever wins my services, I shall argue that their box contains the diamond, so that the box will receive a higher price. And they do that.
And the box owners bid, and box owner B bids higher, so he wins. And then the organizer, the arguer rather, starts organizing thoughts. And he says, first thing he does is write, and therefore box B contains the diamond at the bottom of the paper. And then at the top, he starts writing in all the many factual evidences for why that is. Because there's many signs and portents that, point to one
Blue
box
stamp.
or the other. Yeah, blue
Shiny.
stamp. Exactly.
Lawyers, man.
Yeah, but of key importance is that the clever arguer ignores all the ones that might argue in favor of box A and only writes down the ones that would argue in favor of box B. And Eliezer, yes, exactly. And yeah, Eliezer asks us to imagine a collection of worlds where there is some objective frequency when either box A or box B contains the diamond. If you happen to be an English speaker, you might think that the ink that's on that paper somehow meant that box B contained the diamond. But within the subset of worlds where the ink says box B contains the diamond, there's already a percentage of worlds where actually box A contains the diamond, and that does not change regardless of what's written on the blank lines above. The arguments on the blank line above are not in any way entangled with what's actually in the box. And he contrasts this to someone who's genuinely curious and first writes down all the distinguishing signs of both boxes on the paper, and then applies their knowledge of probability, and at the bottom says, therefore I estimate an 85% probability that box B contains the diamond. Since that one actually has a chain of cause and effect that causes the ink at the bottom of the paper to say 85%, it is entangled with the evidence, and so it will accurately reflect the worlds rather than not being entangled with the evidence at all.
This is hard to summarize.
Yes.
But your effectiveness as a rationalist is determined by whichever algorithm actually writes the bottom line of your thoughts.
Yeah.
Is sort of the summary of it, yeah.
And this post has become pretty famous. Go ahead, sorry. Yeah, all right. But you do... Arya, get out of here. Sorry, my dog. But the thing is, like,
you thought you were talking to Steven.
No, get out of here, Steven, with your logic and words. No, sorry. No, I guess the thing is, like, you do have some evidence. There's conclusions you can draw, but the important part is, like, even if the conclusion is similar for both pieces of paper, like with one of them, the conclusion was written down beforehand, so none of the evidence really matters at all, and that makes a big difference. Like, did you write your bottom line after considering all the evidence, or did you come to it first and then are looking for ways to justify it?
Yeah. This is why it is an excellent idea to publish your planned methods of analysis in advance when doing scientific research.
Yeah, pre-registering your hypothesis.
Exactly. Pre-registration. Something we really don't do enough of in academic publishing, but like we have this exact same issue, right? Where we kind of loosely formulate some hypotheses, gather a bunch of data, and then, the statistical test is definitely the most applicable to my data because of reasons.
Oh. I had gotten the impression that lately it'd become a much more common to do the pre-registering. Is it still not common enough?
Well, so there are specific disciplines, mostly medical contexts for medical trials, where it's much more rigorously enforced. But there's a lot of subcultures within academia, different disciplines, and different norms apply in those different communities. So many still don't have a good norm of pre-registration.
Oh. How do you feel about bioinformatics?
Well, we have a problem, not even with... we have a problem with being able to reproduce our results, let alone pre-register them. I mean, there's a whole rabbit hole that is like another episode's worth of talking that I fear we should probably not go down to discuss academic publishing because somebody restrain me.
Maybe we can have that episode in the future date.
Maybe at some point, yeah.
So it's hard to even get reproductions in bioinformatics? Is that because biological systems are so complex, or what?
Ironically, because computing systems are so complex, we don't have enough emphasis on sort of like reproducible build environments for our analyses, and not enough norms around publication of code and proper source management. It's a whole like most bioinformaticians are trained as biologists and not as computer scientists or as software engineers. So a lot of good practice from sort of industry software engineering is not known to people working in bioinformatics.
That sounds really frustrating.
Yeah, I mean, it's a whole kind of a thing, and also the incentive structure is not great for adhering to those best practices because like the emphasis is on publication, and very few people reviewing publications have the qualifications to consider those computing aspects of it as well. Like they're interested in the methods and the specific biological questions, but not so much necessarily in the process. There's kind of a whole new set of strong norms around reproducibility of code that need to come into force that haven't yet. But it takes more time to do that, and that means it takes more time to publish, which means it doesn't get done.
Yeah, because then you will perish since you did not publish enough.
Yeah.
Damn. Do you think does the new generation seem to be pushing more for that, or is this just a static problem?
There's a few people who there's enough people who are aware that it's an issue, that it's starting to come up. But I think it'll take a while for people to adapt, right? People just need to know about the existence of these tools, and the norms around their use need to get stronger, which has to come from reviewers and publishers. And there's a whole process before the culture changes, but we'll get there.
Yeah.
We'll get there.
With clinical research, it was having software that had to be FDA compliant or compliant with like all of the different regulatory.
Yeah, that's a whole other challenge, regulatory compliance.
Yeah, similar constraints.
Alrighty, let's wrap up this post and then actually get into this stuff.
Sure.
Let's see, where were we? Oh yeah, he brought up the example. I guess Steven said there were some real life examples. Another one was if your car makes metallic squealing noises when you brake and you aren't willing to face up to the financial cost of getting your brakes replaced, you can decide to look for reasons why your car might not need fixing. And then he says, okay, this is really interesting. So I want to talk about this real quick. But the actual percentage of you that survive in Everett branches or Tegmark worlds is determined by the algorithm that decided which conclusion you would seek arguments for. So obviously that's true, but there's a little aside within this, which I found really interesting. When I skipped, he says, but the actual percentage of you that survived in Everett branches or Tegmark worlds, which we will take to describe your effectiveness as a rationalist. which I never noticed before when reading this, but which seems to me really huge. Is this like a good way to decide to describe someone's effectiveness as a rationalist? How many of the multi-worlds they survive in?
It feels like that might be vulnerable to what professional poker players call resulting. Right? There's a similar difficulty in like evolution, right? So if you're only updating based on outcome and not based on method, then in harsh learning environments, you won't necessarily do as well compared to generous or gentle learning environments. Like the difference between chess and poker, right?
Yeah, I mean, but you can't see the outcomes because you can't see other effort branches or Tegmark worlds.
True, but isn't that what he's trying to do
I
here?
think
Like
he
jump
Yeah,
back
you're right.
one level and tot up the number in which you survive.
Yes, you are correct. He is looking at outcomes. Yes.
So that there may be, depending on the environment, specific circumstances in which being systematically wrong, or at least employing some mostly right but less expensive method that doesn't necessarily give you the best result in terms of absolute truth value, might produce better survival results. So you'd end up with a higher total, even if it wasn't objectively the most rational strategy.
Okay, I think I see what Steven's saying, that this is an application for or a description of your effectiveness only in the case of whether breaks need replacing or not as like a general rule. Because like otherwise the most effective rationalist, in quotes, would be the one who never leaves the house because it's risky to go outside your door. Which I don't think is actually going to be an effective life for very many people at all, especially as we now all have, some experience with that over the last year. Yes, but I don't think it is not the most effective way to have a world. I think you have to take some risk in order to, okay,
yeah. That's a similar kind of thing that produces like the origin of like perceptual biases, right? And it's a good heuristic shortcut for getting the answer efficiently and not
not wasting too many resources.
Yeah, you wouldn't like the optimization procedure is not optimizing for like accuracy alone. It's optimizing for survival, which may require sacrificing some accuracy for energy efficiency.
Yeah. And then there's also the question of, is your survival maximized by holding up inside? Probably not, because then society will not get to the point where it can overcome aging, and you're going to die of old age eventually. Whereas if you took some risks and went outside and helped society progress, maybe we could get to there.
Yep,
which would be a great segue to the episode if we were done with this.
Well, that's where the distinction between Darwinian evolutionary processes of the normal kind and mimetic evolution exists, right? That's where, evolution is myopic. It only is interested in what in the past produced successful results. It can't look forward and make adjustments based on models of what it think will happen. It's limited to its statistical summary of the past. Whereas mimetic replicators have got feedback loops, and they can model stuff and have that advantage.
And that's why they took over the world.
Yes.
I was just watching this YouTube video of somebody who is programming cars to run around a track and using evolutionary algorithm to get one that could just drive around the track correctly, and just kept tagging that one, reproducing the one that got the furthest along the track, and it was hilarious to watch because there was hundreds of cars just spinning in circles, crashing into walls, and the one that would just by complete accident end up the furthest along the track would just reproduce that one. And then at the end they did get actually it only took a few generations before they got one that could drive around the track, but it was all they pointed out this is all completely by accident, though. The algorithm doesn't know it's a car. It doesn't know what the car in the track represents.
That's one reason why evolution is very good at producing very lateral solutions, though. Because have you ever seen the analogous visualization of a learning to walk simulation? Because in that one, you get some really weird strategies to solve the problem of get this collection of points to a further distance, right? So there's one where it just makes a really tall thing and falls over.
Yes, that's my favorite one.
It's like, okay, I mean, you solve the problem, but that's not exactly walking.
Good, buddy.
Yeah. Or there was evolutionary circuit design where some evolutionary algorithm figured out how to make a clock without a loop, and it turned out to be exploiting some weird physical property of the chip that nobody had thought of making a clock out of before, because it just wouldn't occur to you.
Yeah, I mean, we don't even have that good of a model of the weird physical properties of the chip, right? We just expect it to run as gates and don't think about, the slight magnetic fluctuations on it as bits flip.
Yeah, we think of it as an abstraction and not as a physical system, and therefore miss the shortcut.
Yeah. The Jace, real quick, the cars, were these like small remote control cars, I'm assuming?
No, it was a simulation of cars on a track.
Oh,
like it was
okay.
just a 2D basically drawing of a track and like the cars, and there was something, I guess, bounding the... I'm not exactly sure how it was like programmed, but the cars could move, and they could only move along the track, and the track was wavy.
Okay, because at first I was visualizing like actual old Oldsmobiles or something, just running into
that
walls.
Would be awesome. Like if somebody programmed a bunch of self-driving cars to try to...
Yeah.
I mean, it would be a waste of vehicles, but it would be really fun to watch.
Like
Demolition
one of those old
Derby.
Destruction Derby. Yeah. That would be awesome.
Yeah. If I ever win the lottery, I want to just fund ridiculous things like that.
I wonder if that would be worth it for Tesla to fund something like that, just as a sort of I guess it already has enough name recognition right now. It wouldn't really help that much for publicity. Yeah.
I mean, you might get some harsher selection pressure on the edge cases, right? Some weird stuff that wouldn't normally crop up in road situations. You might be able to improve the functioning in those edge cases from something like this.
Oh, it would be really great if it was like a whole heap of cars, and one of them learned to shortcut by bouncing off other cars and running them into walls.
Just got really aggressive.
Yeah.
It's like, I got my passengers there safely.
Yep.
Everything else is on fire.
We may have to check.
Okay, does anybody know Elon Musk's marketing person? Because
Tesla, their motto is everything else is on fire.
Honestly, isn't that like kind of the thought behind the family Hummer? It became really popular in the US.
Oh, yeah.
It's like bigger and bigger SUVs.
Yeah.
Brighter and brighter lights blind everybody else on the road.
Till eventually you're running a tanker down the street. Yeah, it was ridiculous. Okay, so to finish up, when he was talking about the algorithm you're using, he says, in this case, the real algorithm is never repair anything expensive. If this is a good algorithm, fine. If this is a bad algorithm, oh well. The arguments you write afterwards, above the bottom line, will not change anything either way. He does say at the very end, as a caution, this is intended as a caution for your own thinking, not a fully general counterargument against conclusions you don't like. The world's cleverest arguer may point out that the sun is shining, and yet it is still probably daytime. So, yeah, people often have a habit of saying like, oh, well, you're just writing your bottom line first to dismiss arguments, which, may be good arguments after all. So remember that this is more for your own use. Do not be doing this in your own thinking and handicapping yourself.
But do look out for other people who might be doing it to try to sell you something.
Yeah.
No, I always thought this had interesting implications for investigative as opposed to adversarial legal systems.
In what way?
In trials, right? I mean, the normal practice is the adversarial court, at least in like descendants of the British legal system, where you've got two sides that advocate for either side, and then there's a few investigative legal systems. I think some of the Scandinavian countries follow an investigative approach where you don't have the sort of oppositional structure. It's more there is an inquiry into the events that took place, but the adversarial system has some merits from the perspective of potential perverse incentives. There's even like the American versus the British approach to the adversarial legal system has some interesting differences, because in the States, you guys have prosecutors and defense attorneys who are always on one side, always the prosecutor, always the defense. Over here we have barristers. They swap. They do both.
Oh, that would be an interesting change.
Yep. Yep. And yep. And and then and it's always that. They always have that perspective on it. They're never on the other side of the table. So
I mean, I like that. I kind of like that system because of, people do just do bottom lining and looking for things that'll support what they already believe. So having two people that have the bottom line of opposite bottom lines, I think will manage to get all the evidence out. But that's only assuming that they're both competent and, interested in doing a good job. But yeah, that it does lead to problems where the people who are always prosecutors tend to form good relationships with the police, and they will sometimes cover for the police, and the police will do them favors, and things end up not being very fair at all if they're in bed like that. Whereas if you had the barristers, then I'm assuming the relationships don't get as cozy. Is that the case?
Well, there's a sort of slightly different set of cozy relationships, but it's less kind of tribally entrenched. It's a bit more. The cozy relationships are kind of with the judiciary, I think, perhaps more. There's, the Crown Prosecution Service has its issues. It's nowhere's perfect, but I think it's probably better than the American approach, to be honest. But
yeah. I mean, even like our comedy shows, I been watching Brooklyn Nine-Nine lately, and they have the thing where all the cops, I mean, it's a comedy show, it's fun, it's lighthearted, but every now and then they go to court and all the cops hate the defense attorneys because they're like, yeah, these are the scum of the earth that are trying to get criminals off of their time and back out on the streets. And that's
creates an unhealthy tribalism. Sorry, another tangent. Yes, it does. Nice segue.
Alrighty. So, yes. Everybody remember those last two segues that we had that were really good, and pretend we said them right now. So, Richard, you are in longevity research?
So the group I work in at the moment is focused on DNA damage repair and aging. And the model system we primarily use is C. elegans, which is a small nematode worm. But before this, I was doing my PhD work in the epigenetics of aging. So I was doing some stuff with epigenetic clocks.
So what does the epigenetics of aging mean? That's the reason that we start aging due to environmental reasons?
Well, to some degree. So back to basics for a moment. You've got your DNA, the conventional double helix. That's your genetic information that doesn't change much through the course of your life. And then you have the annotations to your genetic sequence. That's the epigenetics, right? So the epigenetics are modifications to the DNA itself, which don't change the way that it pairs. That's sort of thing, for example, DNA methylation. It's a modification to the cytosine base that can change the way the DNA is expressed. And then DNA in your cells is packed into chromatin, which is a collection of proteins and DNA. So it's that double helix structure that we're all familiar with from the popular media is wrapped around these sort of disc-like proteins called histones, and they can also be modified. So modifications to those and different versions of those are what you might consider to be epigenetic modifications. So they're on top of the genetic information and provide information about which cell type a given cell might be. So you've got the same genetic information in all your cells. So you have to ask the question of how is it that my brain cells know to be different from my liver cells and so on. And that's what's encoded in epigenetic information.
So it had been my understanding that epigenetics, the epigenetic changes all happen, for the most part, all happen either in gestation or early childhood. Is that true, or am I wrong?
I mean, the sort of the bulk of the epigenetic program, as it were, is laid down in development, and then there are subsequent environmental factors that can influence epigenetic information. There's I think so. Your characterization is pretty accurate. It's mostly it's most plastic to environmental influence early on, and becomes somewhat less so with age.
So, by the time you hit teens, are you done getting your epigenetic stuff modified, or can it still happen?
It can still happen, yeah. It's just that there's like the magnitude, I would say, of the sorts of changes that you could affect would be less. So the sort of origin of epigenetics is actually in developmental biology, because it's epigenetics that sort of lay down the specifics of the developmental program. So you start from a single zygote cell, and then as that cell divides, the environmental cues around it influence what the epigenetic state of the individual cells will be. And there's this sort of self-organizing process that controls what many of those environmental cues for an individual cell will be. So as you develop, there are particular like chemical gradients that might tell which cell where to start differentiating to become an adult cell type. So there's this sort of concept of stem cells and differentiated cells. They're kind of at opposite ends of this axis. You've got the pluripotent stem cells that can become any cell type that you start out with at the zygote, and then you've got the differentiated cells, which are specific functional sort of endpoint cells, like your livers and cells and your neurons. And the developmental program kind of dictates through epigenetic information and through environmental cues, which shape what that epigenetic information will be, the path that a cell takes from that initial pluripotent stem cell down to a differentiated cell state.
So,
okay, I mean, there's a bunch of different mechanisms associated with that, but many of them you can kind of broadly simplify. So, for example, there's this concept of things called morphogens. So it's like a chemical gradient in the environment. So let's say you've got an animal where you've got the head at one end and the tail at the other. And then at the head end, you've got like a really high concentration of some chemical marker. At the tail end, you've got a really low concentration. So somewhere like, I don't know, quarter of the way from the back, you want to start developing some legs, right? So whatever cells are in that area might take a cue from a morphogen that's at about 25% of the maximum concentration at the head end. That says, oh, okay, I'm in the leg region, so I should start going in the leg direction, right? That's a kind of slight oversimplification, but those kind of mechanisms are what yields the patterning of an adult organism extremely. Yeah.
Stuff does happen. Not maybe that.
Yeah, I mean, it's not always perfect, and there are varying degrees in different organisms to which it is defined and to which it is stochastic. Oh yeah.
It is really good. If you remember thalidomide, I think it was that they were prescribing for a while, thalidomide for
morning sickness.
Pregnant women.
That is every single time anyone says anything bad about the FDA, everyone comes back with thalidomide, and you're like, one time they got lucky.
Well, there was also the Tuskegee syphilis experiment, and anyway, I could talk about that. But thalidomide caused, it was a good, like, I guess, pain medication for pregnant women. I'm not actually sure what they were prescribing it for. It caused limbs to not develop in the babiys.
It was prescribed as an anti-nausea medication for morning sickness.
Okay.
And there's the story there is quite interesting because it's there are different isomers of the thalidomide molecule. They have handedness, right? So there's like a left-handed and a right-handed version of thalidomide. And so one of them is toxic.
Safe?
The other one is safe. Yeah. But when you synthesize them, you get what's called a racemic mixture, which is equal parts of both. And they hadn't done enough work in the synthesis process to ensure that they could purify out the right version. And in fact, you can safely use thalidomide for certain things today if you use the right isomer.
Yeah, we're still totally using it in cancer care.
Yes.
Although if you're in cancer treatment, you're probably not pregnant anyway.
Yeah, if you're in a clinical trial, you can't be pregnant.
Okay.
Because of that.
Yeah.
You have to promise that you will use like an effective form of contraception, and you have to describe it, and they have to ask you about it every time. It's really awkward, especially when your patients are like 84, and it's like, so have you been using protection? And it's like, I'm 84 and I have cancer. I am not even sexually active.
Yeah.
The government makes me ask you this.
Yeah, just say yes. That sucks. Also, what doesn't
I wonder
like
if that
chemotherapy and
does that-
radiation? Okay,
Sorry, go ahead.
no, you.
So I wonder, does that have implications for how they do safety assessments for drugs for pregnant women? Because
Oh yeah, it's like a
Must be.
huge debate.
yeah,
It's kind of a between right now. The government went really paranoid about it, where definitely like that you try to err on the side of never giving any kind of drug that have any potential of causing like fetal harm to patients who are still of like reproductive age. But there's another lobby, and it's kind of moving in the direction of, well, it should be the parents' choice whether they decide to enroll in the trial and potentially to like what eyes wide open sign up for it, take something that could potentially harm a fetus.
And
I can
It's
see
kind
how
of
the
moving more in that
conservative
direction
approach.
now. Last I heard, anyway.
I could see how the conservative approach would have better PR value.
Yeah.
isn't everything potentially harmful to a fetus? I mean, that sounds a lot like California's. This has been found by the government of California to potentially cause cancer thing.
Oh well, yeah. No, it's like unless it's there's like various categories, and there's a few things that have been approved, but like very few. And if it's a research drug, then like you just don't know. So
Yeah.
it excludes most things.
So to return
But,
a minute to the
yeah.
the developmental biology stuff we were talking about before, there's a useful image I'd like to put in people's heads as a way of thinking about developmental biology. So there's this guy called Conrad Waddington, who has this notion of the epigenetic landscape. So if you picture a hillside with a valley running down it, like you start with a single valley at the top, and then it branches out into many tributaries, or not tributaries, branches. Tributaries would be the other way around. Anyway, yeah. So if you then place like your pluripotent stem cell at the top of this landscape and kind of picture it rolling down the slope, you have a bunch of places where you can pick where it'll go into which branch of the river. And those are like the developmental decisions, right? That's where something changes in the epigenetic state of the cell, which says, I'm not going to be in this cell lineage, but I'm going to be in this other cell lineage. And then once you've rolled down to the bottom, you've got, so each of the different valleys that the rivers run into represents like a differentiated terminal cell state.
And so what does all this have to do with aging?
Yes. So good point. We should bring it back to that. So there's kind of there are many theories associated with what aging is. There's a lot of sort of damage-based theories like reactive oxygen species, accumulation of mutations. But one that's kind of gaining a little bit of popularity at the moment is a sort of epigenetic information theory of aging. It's quite popular with a guy called David Sinclair, who's pretty well known in the field. He's written a popular book on the subject. I think it's Why We Age and Why We Don't Have to is the title, which is a bit of an overstatement in my opinion. But you got to sell books.
We mentioned him on our last podcast too.
Yes, he's well known. There's a couple of he's very slightly controversial on a couple of points. Just he's a solid scientist in most regards, but he has a slight tendency towards hyperbole in places. That's just So
I mean, I also would like to not age, but I don't believe I can totally stop it yet.
yes. I think it's a slightly more thorny problem than he at the moment characterizes as. Actually, the developmental biology picture is a really good place to start on where I slightly differ with David on this. One of the so if we return to that picture of the epigenetic landscape for a second, and we've got these cells in the valley at the bottom of this hill. So when they're functioning well, when they're doing their job correctly as a terminally differentiated cell, it's a liver cell being a good liver cell doing liver stuff, right? If it starts to become less liver-like, either by maybe sort of jumping over to the next valley a little bit, or rolling back up the hill ever so slightly, becoming a bit de-differentiated. That, I think, those sorts of properties are what we see in aging cells, right? They cease to become quite as tightly functional in the role that they were sort of differentiated to be.
They can undifferentiate?
Just a little bit, yeah. So what the differentiation is kind of enforced by the epigenetic information. So the particular state of the epigenetic information in the cell tells it what genes to express, what environmental stimuli to listen to, how to react to stuff in the environment. So if that information over time becomes degraded, as information does when there's a finite amount of energy to maintain it, you will lose the precise description of that state, right? So it becomes slightly less livery than is ideal for a liver cell, right?
Oh my God. I so I love this metaphor because like as I've gotten older, I found myself not giving as much of a fuck about like external
Nice.
pressures in my environment. I'm like, yeah, I don't really care all that much what you think. I'm okay. I'm set in my life, and I'm just gonna ignore certain particularly whiny people. But like now, I'm viewing all my cells as being like, yeah, I don't give a fuck about what you think. I'm not gonna be that much of a liver cell anymore.
Yeah,
I'm like, so my cells are literally becoming grumpy old men.
yeah, pretty much. And occasionally
God
they have
damn
a midlife crisis and go off and become cancer. That's
Oh, there's nothing worse than old people. Jesus.
yeah. But so
Apologies to any older
so
listeners.
to return to the point where I think David's a little bit off about this. So what? So his sort of epigenetic aging hypothesis kind of has this idea of if you take these terminally differentiated cells and you roll them back up the hill just a little bit, you de-differentiate them a touch by using some of the factors that you find in stem cells, and then let them roll back down the hill again, so they'll kind of settle back into the groove.
You could do that?
That's the idea, right? So the idea is that you de-differentiate them just enough that you can get them to start doing what they should be doing again. But the catch is that the process of deciding in which valley you end up is determined by those developmental cues. So to go back to what we talked about earlier in the conversation, that like gradient between like the head and the tail, where you had a particular concentration that was close to where the legs are, so you should become legs, right? That information, that chemical gradient, is present during development, but not in adulthood. So when you roll the ball back up the hill, it doesn't necessarily know into which groove to roll back down, because there's not the environmental cues present to say you should be going this way.
And you can't give an adult like a chemical cocktail to stimulate that?
Well, that's where it gets a little bit more complicated, because it's a very precise chemical cocktail that defines what particular function a cell should be doing. And so the idea in this sort of epigenetic rejuvenation stuff, where you roll a ball back up the hill a little bit and let it roll back down, is that there's enough information in the environment and in the cell's epigenetic memory to remember where it should be rolling back to. But I don't think there's quite enough there to get it to reliably roll back into the right spot. I think we need something like that chemical cocktail that you described to ensure that it'll end up back in the right spot instead of becoming cancer.
And we don't have anything even approaching that chemical cocktail yet.
We have something resembling it in some areas, but delivering it and the specifics of how we might go about it, that's still very up in the air. There's a lot of people in the field of regenerative medicine who are working on figuring out how to cause a cell to differentiate into a particular type of cell. So if you fuse together those two approaches, if you take this notion of roll it back up the hill just a little bit, and then you add to it some direction with some chemical information of where you want it to go, then let it roll back down the hill, that might work. But at the moment, those are somewhat disparate areas of research, and they're both pretty early on. So combining them is sort of the next step, I think, in both of those areas.
Okay. How do you roll it up the hill?
So that's what is usually used there is some subset of what are called the Yamanaka factors. So those are a set of genes which induce pluripotency, so that ability to become any other cell type in an adult cell. So you can take an adult cell, you can express 4 genes mostly, and then it will take on the properties of a stem cell. Well, some of them will. There's a success rate. Not all of the cells end up becoming pluripotent. But
How do you
so
how do you
go
do
ahead.
that though? Do you like have to directly inject each individual cell, or
Well, it's often a sort of viral transfection method where you might put the genes in some genome engineering vector in a virus, and it kind of gets pasted into the genome of the cell you're looking for, or it might be expressed separately on the vector. So instead of integrating it into the host genome, you just have it present in the cell so it can express those genes but not be integrated into the host genome. There's a bunch of different specific methods of how you might do that, but some form
but
of...
that,
Go ahead.
but that's like individual cells, like on a petri dish or something, right? Is there some way to do that in a human body?
Well, yeah, only with at the moment the most successful methods are using viruses to introduce the genetic material.
Okay.
But the success rate is a bit low. So only one in, well, it varies a lot by experiment, but one in some number of cells, might be 1 in 100, might be 1 in 4, if you're really lucky, will end up with a copy of this genes that you want to express.
I don't know if anyone's doing this, but with cancer care, we were taking CAR T cells and harvesting cells from the patient and artificially just reproducing the ones that had the programmed instructions to kill cancer cells. So you'd and then put them back in the person. And then there's a newer version where you could take donor cells and do the same thing. So I have no idea though if
I mean that's
definitely
definitely
used outside of cancer care.
So I think that like blood's kind of a unique system in that regard. That you can take out something that's going to be a blood stem cell, select for it, and put it back in the blood stem cell niche, and then have it produce new blood cells. Most other tissues don't function in quite that same way, so it's a much more challenging problem to do the selection outside and put it back in, because you don't have that same the same way the tissue works with respect to the production of the blood cells from a stem cell population. Right? There's not the same turnover rate. So, for example, you can't really do it with brain cells because like each individual one.
You don't want to replace all of your neurons.
Exactly, right. If you take them out,
kind of
mix them
the
up
purpose.
into a soupy like substance, and select the ones that work, and then put them back, it's not going to work.
Would I really want all my cells to become pluripotent at once anyway? Because,
no, that would be very bad.
Right. So
have you seen the movie Acton?
Yeah, right. So 1 in 100 sounds pretty decent as long as I could keep doing it for all 100 times.
Yes, and so that is one of the approaches that people are taking in this kind of therapeutic area is like inducing them permanently can be an issue. It's the whole sort of cancer thing. So having a small number of them turned on periodically is an approach that might work. So you can put genes on a in like an expression cassette is what they call it, where you have a gene that will only turn on if you're taking a drug that will activate the transcription sites that cause that gene to be taken. So you can take an innocuous chemical compound that will turn on a particular gene expression cassette. Now, so they have really great results for that in mice for eliminating senescent cells. So some of those cells that do that slightly dedifferentiation thing, they kind of end up not quite being as livery as a liver shell should be, right? Well, it's a little bit more complicated than just that, but that slightly dedifferentiated cell can end up being a senescent-like cell. So the senescent cells have a particular phenotype. They secrete a bunch of stuff that's kind of cytokines and inflammatory things that cause increased inflammation in the tissue, and they are slightly prone to inducing senescence in other cells around them, and they're not really doing what is functionally useful in that tissue. They have what is thought to be kind of an anti-cancer property, so they sort of, it's a pseudo niche that a cell can become. So instead of being dedifferentiated and becoming cancer, it can dedifferentiate a little bit and become senescent. So it's a kind of a purposeful pathway.
Like a placeholder cell.
Yeah,
almost.
it's kind of a little plate. It's like, do this. It's not as bad as this other thing, and it might help a little bit in some ways. So those senescent cells and their SASP, senescence associated secretory phenotype, which is the jargon, the cytokines and stuff. Yeah. Yeah, they like their acronyms. Is producing inflammation. So that has implications for your immune system as you get older. But if you target those cells to eliminate them, so one of the things they did was they genetically engineered mice with one of the markers, so the genes that are expressed in senescent cells. And then once they reached adulthood, they gave them this drug, which caused all of the senescent cells to sort of basically just kill themselves off. So what you end up with is a mouse that, as an adult, can be induced to suddenly lose all of its senescent cells.
Oh my God, that sounds brilliant.
Yeah, and depending on sort of how you administer it, it's usually through some kind of programmed cell death, like autophagy, just induces that process in the senescent cells. Although sort of one of the properties of senescent cells is they're not great at apoptosing, so you can kind of force majeure it.
Seems like it would be really traumatic for the mouse, depending on how many of the cells were senescent.
Yes, and I think the way that they, I forget the exact regimen they use. I'd have to look some of this stuff up. It's not really my niche, as it were. But they, I think you can dose the drug, the doxycycline is what they usually use, over time. So it's not sort of one acute, all the senescent cells die now. It's a bit more gradual than that. But if you look at mice that have had this done, it's night and day, right? There's like a perfectly youthful looking, brown, smooth coated, bright eyed, perky, straight spined mouse. And then next to it is like a gray, bent, kyphotic spine, just generally decrepit looking mouse. And yeah, it's a really, it's amazing. And the same age. Yeah.
So, how long can this go on for? Like, can the mouse just keep doing this indefinitely?
Well,
not indefinitely, but I think
not indefinitely. But
they've increased a mouse lifespan to like 10 years, right? Where normally it would be like 3 years.
I don't actually know what the sort of record is for mouse lifespan, and it would vary by strain. But yeah, there are interventions that have seriously extended mouse lifespan by quite a considerable amount.
I have two questions about this. The first one, kind of sillier. Does the mouse get smaller each time?
No, actually. Although weirdly, the one that's healthier looking is quite a lot bigger physically. Huh.
So those senescent cells get replaced by something else that's equal.
And I think that also there's less. It's a very good question. Come to think of it, I'm not. Yes, they're also they're post-mitotic. They're no longer dividing. Yes. Yeah, that's a sort of self-destruct. Yeah. No, just and sort of more average-looking size as opposed to exhibiting frailty. Right, as we get older, we get kind of thinner, and skin gets looser, and bone density drops, and all that unpleasant stuff. Yeah, it's not like
how old people are because they're like the biggest, awesome people around.
No.
They're like lobsters. They don't get older, they just get bigger.
There is an interesting paradox relationship there, actually, with size of organisms. So generally speaking, the bigger an organism, the longer it lives. But within species, smaller individuals of a given species tend to live longer.
Yay.
It's a weird thing. But
I'm like 5'2".
Damn it, I'm going to die soon. So my second question, and I think the far more important one, is, can this be done in humans too?
The mouse one with the sort of senolytic thing where they break down so the particular mouse approach is not really applicable to humans because it requires genetically engineering from birth with this expression cassette.
Well, can at least our children be made this way so they have better lives?
In theory, if we go ahead with the whole genetic engineering of our kids thing, which is a whole other thing. Yeah, and assessing the safety of that is so that's a whole other thing. I think we should probably pursue the whole germline genetic engineering route as a species, but that's another debate.
Like the safety arguments really get to me because if
Hmm.
if you don't pursue it, you're gonna die anyway. The lethality rate of a typical human is 100%. So there is no safety argument.
Right, you don't want to accidentally genetically engineer kids that will die at 6.
Yeah.
Like,
Sure,
that,
but
that would be kind of a tragedy.
I mean, yeah,
We're
that
trying
would be
to avoid
terrible.
that happening.
But like, if you die at 66, that's also a tragedy. So you're gonna
But,
die
yeah,
either way.
Yeah.
I mean,
at the very least, society regards it as less of a tragedy.
Man, society's a load of crap. So right now, there's no way to reverse
So,
engineer them into living
Actually,
humans.
this is one of the most interesting areas of drug development in the anti-aging space at the moment. So senolytics, which is a class of compounds that will cause senescent cells to be broken down as a drug intervention. A lot of those are being funded as investigations at the moment. So there's sort of some preclinical work and stuff going on with a bunch of startup companies. There's
Oh my
a
God,
lot of
can you get
Silicon
me-
Valley people interested.
Can you get me into one of these trials?
I'm afraid I don't have those kind of connections.
Oh, damn it. Okay.
Not
if,
yet,
if any of
anyway.
our listeners can get me into one of those trials, I will be your guinea pig.
Yeah, but that's definitely an area that people are very much looking into, and there's a lot of diversity of senescent cells. There's a lot of debate around the precise definition of what they are. There's different sort of subtypes, and successfully targeting senescent cells so that you have a compound that will only kill senescent cells is kind of analogous to the cancer cell problem, right? You need something that will only kill the cancer cells, you need something that will only kill the senescent cells. But in the senescent cell case, you have less of a tolerance for toxicity because you're dealing with otherwise healthy people.
What the hell makes cells get senescent in the first place?
Yes, so that's an interesting one. I mean, like, if you want to get only slightly facetious, and that's a hard word to say, facetious, then the second law of thermodynamics.
God damn it.
Like, kind of thing. So there's this interesting situation we have where we're an energetically open system, right? So theoretically, that's not too much of an issue because we can take energy and we can use it to do stuff, including repair things.
Mhm.
But there's an interesting sense in which we're an informationally closed system. So here, I find it useful to talk about Nassim Taleb's coinage of anti-fragility. So you've got this notion of anti-fragility, which is something which is made stronger or better by stress in the environment, right? And then you've got robustness, which is something which is only resistant to damage. And then there's fragility, right? So, and the metaphor that he uses is the sword of Damocles is fragile, a phoenix is robust, and a hydra is anti-fragile. Because you cut off a head, it grows another one. So the genome
grows 2 more.
Sorry?
If you cut off the head, it grows 2 more.
Yes, 2 more. Actually, yeah, getting actively stronger. So as is usual, we have to talk about evolution a little bit now because, as Theodosius Dostoevsky said, nothing in biology makes sense except in the light of evolution. So in a population, the genome is anti-fragile because there's a feedback loop between what the genome causes to be the phenotype and what the genes end up being, right? So that's the process of natural selection. So the genotype causes the phenotype, which is what the organism is and looks like and does, and then you've got the consequences of that in the environment, which is where a natural selection acts. So if a gene has a consequence which is good for the phenotype and therefore is selected for in this particular environment, you've got a mechanism that can correct what the genome looks like. It can undergo what's called purifying selection or other forms of selection, right? So you can, in the specific case of fixing something that works if it gets broken, like in maintaining state, that's purifying selection. Right? So an individual, on the other hand, doesn't have that. Right? If an individual, you're only robust, you're not anti-fragile because there's no connection between what your genome is doing to the environment back to the genome.
But I mean, I can see the whole culling and reproducing to drive evolution thing happening, but wouldn't that work just as well if the environment culled people due to them becoming too unfit for the environment rather than just having an expiration date on every individual?
So there's slightly different things that I'm talking about. So the point that I'm trying to make is there's not like for me as an individual, there's not a feedback loop between what my genome is causing my body to be and what my genome is.
Right.
But if there's a population of me and there's a selection effect going on, there's feedback that connects what my phenotype is to what my genotype is.
Mhm.
Um,
Yeah, I agree,
Hmm.
but I mean, your phenotype or your population would keep getting stronger through just the natural process of culling and replenishment, even if like the strongest one from the last generation wasn't automatically killed for no good reason.
oh yes, yeah. But my point is that just that me as an individual, I'm robust. My genome is robust. It's not anti-fragile because because there's not that feedback loop, which means that if my genome starts going wrong, right, if I start to accumulate some mutations, I don't have there's not like an external point of reference, right? I can't go to something else to appeal. What should my genome look like?
Yeah, you can't factory reset.
Exactly, right? So there's only so much redundancy, right? You've got 2 copies of your genome and all your cells, so you can, if you get some mistakes, and there's a whole bunch of really impressive and complex mechanisms for repairing DNA damage, but that's all they can do, right? They can repair the damage. They've got a sort of finite set of information with which to work. So when you think about it as a physical system, it's kind of an information transmission problem, right? You've got a set of genotype information that you're trying to send forward in time, and like the physics of signaling mean that there's a sort of finite amount of energy that you can expend to maintain state, like at a certain probability, right? So if you want to maintain the state of a particular bit, right, if you want to know that this bit will always be 1, then you have to expend a certain amount of energy to maintain that state with a given probability for a certain time horizon, right?
So
So
it's
that's
primarily that the genome degrades over time beyond the ability of the cell to be useful still?
it's a little bit more complicated than that, because it's not like we're accumulating so many mutations that everything starts sort of breaking down. And that's where the epigenetic information theory also sort of comes back in, because it's not all of the cell's information is in the genome. A lot of it is, and relevant parts of it are, and there's definitely a role for genetic damage in aging, like because DNA repair mechanisms come up over and over again in premature aging syndromes. So these progeroid diseases where individuals age really rapidly, a lot of those rare genetic diseases have mutations in DNA repair mechanisms. So DNA repair and maintaining DNA state, definitely important. But there's also implications for the epigenetic information. So even if you've not accumulated enough mutations for that to be catastrophic for your body functioning, you may have accumulated enough epimutations. So back to what we talked about earlier with the epigenetic information sort of encoding the information about what state a cell should be in, about maintaining a particular differentiation. If you like, the same physical principles apply as to the genetic information, right? If that gets degraded, if there's some loss of information because there's a finite amount of energy you can expend on maintaining it, then you start to get that dedifferentiation phenotype.
Okay. So it's mostly about the epigenetic information getting degraded over time.
It's kind of a mix of both. We don't really know to what extent it's epigenetically mediated, to what extent it's genetically mediated. It's actually very challenging to characterize like low frequency mutations, like how often they occur in somatic cells in the adult organism.
And it's more than just the genome, right? Aren't there, I don't know if you agree with the like Aubrey de Grey's 5 factors of aging or however many factors there were, but like
yes, yeah,
it's a bunch of systems breaking
there's
down.
The whole hallmarks of aging hypothesis. Yeah, I mean, so all of that, I think, to some degree, links back to this sort of information problem, because you start getting feedback cascades, right? So once you lose a little bit of let's say you lose a bit of genetic information, right? So you have a mutation that affects your ability to repair DNA. That means that you have a higher probability of subsequent mutations because you've got an impairment in the repair process. And the same thing can apply in other systems like epigenetic systems. So if you get a point mutation that affects your ability to accurately continue to maintain the epigenetic state, then that can begin to become corrupted over time. So you get these positive feedback loops where you lose one piece of regulation of one system, and that causes some other system to no longer function as well at maintaining its state, and so on.
Yeah, and then so some of what causes aging is sort of your body trying to mitigate this damage to extend the lifespan rather than
Yes.
The
Yeah.
example of the senescent cells, it's like a jury rig
Yeah.
to keep the organism going.
And
And
it also comes back to the evolutionary question again, because there is a certain amount of selection for a given lifespan, or at least a sort of minimum degree of functionality at a point in time, right? So, I mean, evolution's utility function is to maximize for inclusive genetic fitness, right? That's what it's sort of grinding to do.
Or reproductive fitness.
Reproductive fitness, yeah. So if your reproductive fitness is in some degree orthogonal to your longevity, then it will optimize for the reproductive fitness and not the longevity, right? Longevity is not necessarily coupled to reproductive fitness. So evolution only cares about it to the degree that it is. So there's definitely a degree of coupling, but they're not perfectly coupled. They're not perfectly aligned.
Yeah. Like there's the thing where social species, evolution let humans or elephants live long past reproductive a reproductive age because having grandparents increases the genetic fitness of the offspring.
Absolutely.
And the offspring's offspring. But with the example of elephants, it just keeps the elder elephant around long enough to ensure that the grandchildren have a better chance at life, but their teeth fall out.
Yeah.
And they just slowly starve to death. It's horrible.
It's that, and humans have this too, because having grandparents is super beneficial for us as well as for elephants.
Yeah. We can talk. I mean, elephants can sort of talk, but we can, tell the history of our tribes and explain, what to do in case of a war or where I hid my cache of gold.
That's where it can get really interesting looking at model systems that are short-lived. So there's a really interesting collection of species called African turquoise killifish that live in seasonal lakes. So they have the whole life cycle in a series of weeks in the rainy season. So they hatch, basically in a puddle, right? And there's kind of these seasonal lakes that are sometimes bigger, sometimes smaller. And there's a bunch of these related killifish species that have slightly different lifespans that are tuned to how long the water lasts in their particular set of lakes. So you get this effect where if you don't need to live long enough to, if you don't need to live longer, then evolution stops caring about the other stuff. There's this notion of the selection shadow, right? So after you've reproduced, evolution no longer cares about whether or not you're functioning, unless that has an impact on the reproductive success of your kids, right? It's the weird caveat for the oddballs that are elephants and us, right? So there's this kind of use it or lose it thing in biology where if those genes are not being selected for to do this maintenance, then if we can get an edge in reproduction out of changing them, or if we can just save a bit of energy so we can do more other stuff that's not relevant to living longer, right? So maybe this gene has an effect that would cause you to live longer, but if you can tweak it a little bit to be slightly worse at that, but it doesn't matter because you're going to die because the flood's going to be over.
But you can have more
save
eggs.
energy to have more eggs. Yeah. So you get this evolution will cannibalize longevity in a situation where it's beneficial to spend that energy somewhere else because you have always got a finite energy budget.
So what I'm hearing is that this is all my ancestors' fault for not continuing to fuck well into their hundreds.
Yep.
That's one way
the strategy that tortoises use. There's the Galapagos tortoise that lives. How old did they live? Very long time.
Yeah, some absurd number of hundreds of years.
They stay. They continue to be fertile, right?
I believe so. Yeah, for quite a long time.
There's long-lived species that I don't get, like parrots or whatever. Maybe that's just random. The evolution didn't cannibalize those longevity genes.
Presumably, there is some environmental factor that makes that work for them.
Yeah, parrot's a bad example because that's also a social species.
Yeah.
Trying to think of something else though. I know there's immortal clams and stuff like that, but then we may as well just be comparing alien species.
Well, I mean, the clams presumably get some selective benefit from living as long as they do. There's this other concept that's quite useful called antagonistic pleiotropy, which is kind of an unnecessarily complicated name, but it's antagonistic, just against antagonists, and then pleiotropy means to have many functions or multiple functions. So a gene which has a beneficial effect in the short term for reproduction, but a cost later on in later life, can be selected for because evolution isn't seeing that cost in later life effectively.
Right.
So that can mean that you have selection for stuff that will be detrimental later on, but happens to work out well early on.
Okay, so maybe in theory we could be much less fertile in youth and live to be much longer, but evolution never saw that thing as useful.
Yeah, that
Okay.
might be
This
a
is why I have
trade-off.
this pet peeve about how the pop idea of evolution always sort of almost substitutes it for God. Like, and the power of evolution, like they like characterize it as this beneficial force. I'm just like, no, evolution's terrible.
Yeah, that's like
The system of non-random death.
Red in tooth and claw. Yeah, it's not pretty, not at all.
So earlier you mentioned that senolytics are being worked on, like drugs that would target senescent cells and rejuvenate people that way. How far out is that? How are things looking? Do you know anything about that?
So the senolytics are still pretty early, as far as I can tell. I'm not super current in their development. I can probably point to a few news sources if people are interested, where you might get more current information about develops there. But there was a preclinical trial that didn't go terribly well, aiming at treating, I think it was arthritis in the knees with a senolytic compound. But I don't think that should necessarily be read as being a serious problem for senolytics. It was a small early trial, and it more of a safety thing. I think the efficacy could easily be better than might be suggested by that.
So didn't go terribly well. You mean it didn't do what it wasn't supposed to, or you mean it like blew up people's knees?
Didn't do what it was supposed to.
Okay.
I think from a safety perspective, it was, if I'm recalling correctly, it was fine. I should probably, if we're entering into the discussion of drugs, so I should probably just I am not a medical doctor.
Right.
Do not take anything that I say about longevity interventions here as medical advice. If you do anything to intervene in your sort of medical regimen, do so in consultation with your physician. Do not take advice about it from random people on the internet, like myself. Right. That said,
do you know do you have what would be your guess as to how senolytics are coming along? How many years or decades or for something initially functional?
I would not be surprised to see some senolytics on the market for specific treatments, like something like arthritis, in the next 5 years or so.
Holy shit.
Something, I mean, that's kind of a moderately optimistic timeline. But for senolytics, particularly for general purpose longevity intervention, I think that would be a longer time horizon, more in the 10 to 20 year window. And again, the effect sizes are not necessarily huge, right? We don't know how much of an impact on lifespan they have, right? So even if it's a small one where you're seeing a couple of extra years on average, they might start using them as longevity interventions. But it depends what your degree of interest is in marginal gains, as it were. Right? Similar with a lot of the other longevity interventions or supposed longevity interventions that are on the market or that are popular by people in this space. So there's a few that, you mentioned in the last episode, NAD boosters, NMN and NR, those are pretty popular. They seem like just a priori, those are the sort of things you'd expect to be pretty safe because that's a very prevalent molecule in the body as is. It's a very it's fundamental to metabolic processes. So from a safety perspective, I take NMN supplements. I think they're probably safe. As to how likely they are to actually be effective at lifespan extension, that's yet to be borne out by a good systematic study. Other popular ones are metformin and rapamycin. Metformin is normally prescribed as an anti-diabetic, drug, or as a drug to control diabetes. But there's some reasonable evidence that that has an effect on extending longevity. I mean, it's relatively small effect size, but it seems to be there. There's pretty strong evidence for that in mice, and there's work in place to have a large-scale human trial on that one because it's metformin. Yeah.
Alrighty, that's going in my black market shop list.
Do a lot of research on it because it has adverse effects that you might not find to be the cost benefit analysis.
Absolutely, yeah. And I made some notes before the show. I can send you a couple of links for reference. But this is one of NAD's probably fine. That's the one I take because just a priori I can't see much that's likely to go wrong there. Metformin and rapamycin, slightly different stories.
What are the side effects? It sounds like they're not good. Well, it sounds like they're extra bad from the hesitancy I heard.
So metformin, I don't think is it. I don't know much about the specifics of the side effects of metformin, to be honest with you. I don't think.
Jace, did you have opinion?
Did you? Yeah.
So it increases lactic acidosis. If too much metformin accumulates, you could get chronic or acute kidney problems, or like liver problems, or even heart failure. Which that's like all worst case scenario things, but.
Well, getting heart failure from trying to not get old sounds like a bad compromise.
Yeah, although the people who advocate for a lot of this stuff will make the case that getting old is the single largest risk factor for heart failure.
That's true. Yeah.
So that's and for the most part, those risks are like for a lot of drugs. The added risk profiles are like for kind of rare edge cases, right? A lot of people won't experience many of the specific side effects. Sometimes the side effects are very prevalent, but something that's as severe as heart failure is unlikely to be a very common side effect of a drug that is in common use. And metformin is used a lot, right? A lot.
The stomach
of
effects are a very common side effect, though. So
yes.
So you have to trade off, the potential to live 2-3 years longer than you might have, with just having chronic diarrhea. And other kinds of stomach disturbance. Again,
there's a lot of individual to individual variability. Some people will have very limited side effects. Others will have quite severe ones. It's dependent on people's individual genetics and environment and microbiome, all kinds of stuff. And rapamycin is perhaps the most controversial potential longevity drug. Some people make a very strong case for it. But it is used.
I think that the mouse that won the Methuselah Prize, they used rapamycin.
Yeah, it is. It's one that has a pretty reasonable effect from what we can see. But it is an immunomodulator and is used in immunosuppression in some areas.
So extra bad during COVID times.
This is the thing. It's not necessarily. So it's an immunomodulator, not an immunosuppressant as such. So there's the specific, I think it's transplant.
It's used for
where it sort of reduces the risk of transplant rejection. But there are other cases where it might actually improve immune function in certain areas. So it's not a straightforward just dial down all immunity drug. There are more nuances to it than that. These are the ones that, for example, David Sinclair has said before. He takes all three of those as a cocktail.
How did he get prescriptions for them, or is he also sailing the high seas?
I don't know exactly, but he always talks about having a sack of rapamycin in his basement that he just takes a tablespoon of the stuff. It's
I don't know about rapamycin, but I think metformin is pretty easy to get.
Yeah, metformin, relatively speaking.
Drug.
Yeah, if you do analysis on river water, you see trace amounts of metformin. That's how commonly it's prescribed these days. It's all over the place for diabetics.
Okay. I guess all you got to do is knock over a rapamycin bank one time, and you're set for life.
Yeah, somehow get a hookup from David. Who knows?
So, what exactly do you do in the research field in here? Are you working on directly with the Senolytics company
No, I'm not doing any corporate work. So my PhD work was one of the things I was looking at was trying to create epigenetic clocks, which actually is a thing that's worth talking about because
Yeah, tell us, what is that?
so an epigenetic clock is effectively just a means of estimating how old you are based on the state of your epigenome. So you take a machine learning model, or it's a pretty simple machine learning model for those interested. It's just, it's barely machine learning. It's they usually use elastic net regression, which is sort of a compromise between ridge and lasso regression, to pick a set of estimators. So a set of, in the case of DNA methylation clocks, particular CpG sites, which is where the methylation takes place. You look at the amount of methylation that's present on a collection of sites, and that gives you a very accurate prediction of how old someone is. So if I take a measurement of the methylation state, and then I run this predictor on those numbers, I will be able to pretty accurately predict how old you are, or at least how old an average of a population is. Like how good it is at individuals is a bit variable, but you've got a very good estimate for the average of a population age.
Have you taken the test yourself?
No, I have not. Yeah.
Oh, okay.
No, yeah, it would be fun to do. Yes, yeah. In order to actually create one of these estimators, you're doing usually a DNA methylation assay. So you take some blood. The DNA methylation assays use something called an array technology, which is originally used for doing genotyping. So you can treat the DNA that you extract from an individual with a chemical process involving bisulfite that will change all of the non-methylated seeds so that they will pair differently. And then you run this array and you can see whether or not when you try to incorporate a new nucleotide, if it's changed. So you can infer whether or not a site has a methylation or not. So with that, and each of these probes at a site has a particular location in the genome. So from that information, which uses a fluorescent probe, so you kind of shine a laser on this glass slide with a bunch of little dots on it, and each one, depending on the color, tells you whether or not the particular location in the genome is methylated. So then you have this big list of sites in the genome methylated to a greater or lesser degree in a given tissue. So with that big list of like numbers, but just sort of a proportion that's methylated, you use that as your input to the machine learning model, and the age of the individual from which you took it is the thing you're trying to predict.
And does people's like methylated age differ often from their years on Earth age?
That's where it gets interesting. So whilst it's very accurate at predicting your chronological age, the difference between the predicted age and your chronological age is very informative about your biological age. So if you are older in methylation age than you are chronologically, then you will probably die sooner than if you were younger.
That's a bummer.
Yeah. Plus or minus 3.5 years-ish. Yeah.
Is it?
So we don't, there are certain things that modulate it a priori. So like smoking, you smoke, you have an older biological age by the methylation clocks, which is unsurprising. And there are a few kind of environmental factors, but smoking, I think, is by far the largest. We don't really know that well what the things are that modulate it. That's kind of an active area of research. But one of the challenges in doing longevity intervention research is it takes a while, especially in humans, right? So, so if you want to test whether or not an intervention works, it's impractical to give it to people when they're young and wait till they die, right? So the good high quality biomarkers of aging, like the DNA methylation clock, are a great tool potentially to use in that context. So if it's a good proxy for longevity, then if you give someone a treatment and you see their biological age according to one of these clocks come down, then okay, maybe this treatment will work to extend longevity. So that's one of the spaces where these are looking at being used.
The NMM and
NAD
nicotinamide riboside. Why is it easier to say than the acronym? Anyway.
Yep. Yes, absolutely. Yeah, there have been a couple of early trials of this with small numbers of individuals with longevity increasing interventions that have shown promising results for this as a proxy. So it looks like this will be one of the things that is used as a tool in the longevity extending drug development space going forward. Well, so the ones that have been done so far are mostly the sorts of things we talk about before with like metformin and rapamycin, NAD, that kind of stuff. I forget which. There's a small trial with like 10 participants where they did something along these lines, and that showed a small effectiveness, but it's so small as a trial that it was kind of a proof of principle. It wouldn't really say anything strong about the actual efficacy. So when they go bigger, it'll be interesting to see. But the other thing is they do know this works in model organisms to a degree, because they've done this where you can show even in like the C. elegans worms that they work on in the lab where I am now, that treating with longevity extending interventions shows decreases in biological age as indicated by a clock. In the worm's case, it's a transcriptional clock because they don't have DNA methylation, but same principle.
Is there any way to get one of these methylation tests, like a 23andMe type service, or?
Actually, I believe there is, yeah. Although I'm not sure I'd recommend it.
Oh, you don't think it's very accurate?
There's conditions that could cause poor methylation, and it could tell you if you have one of those. I don't know if it's like you'd have to pair it though with this machine learning.
That's actually, that would be very easily done if you got the raw data. Right? It's basically a quadratic equation with a fixed set of terms. Well, not quadratic, but a polynomial equation with a fixed set of terms. Right? It's you're multiplying by some defined set of numbers that's published in these papers and getting an output. It's super simple to.
Oh shit! I'm going to do it.
The model.
Well, I mean, you wouldn't recommend it because the commercial ones aren't very good right now, or
so. I can't really comment on the quality of the commercial ones. There's no particular reason why they would be good or bad. We still don't know very well how good a predictor of individual difference this is, because like we've been doing it at kind of population level. We say like the average in these 100 individuals is that this many are accelerated, this many are less. We don't really know how effective it is at like the individual level. I mean, it might be it might be an interesting thing to have, but but if you got some $500 or whatever it is to burn on the test, then maybe. But the other thing to bear in mind is the privacy and data element of it, because there's a decent chance that if you go for whatever commercial offering there is there'll be some terms and conditions that say we can use this in however various ways we wish. Like 23andMe has many of those catches. So it's depending on your feelings about giving the company information that might potentially one day impact on your ability to get life insurance. That's.
But how could they tell like what your cellular aging is if it was above or below the norm, if they couldn't tell what it was within the 3-year margin?
Well, I mean, you still get so that's a source of noise there, right? Yes, I mean you get an exact predicted age with some error bars, right? So we normally just effectively ignore those error bars, but somewhere in the error is a mix of technical error and biological age information.
So do you okay, and I was wondering, do you feel like comfortable giving a name of any service, or is this something you'd rather just not and let people know themselves?
If I couldn't actually remember any of them, I think there's one called Elysium, maybe?
Elysium is the company that makes NAD supplements. Wait, no, then that's, and they also...
Oh, they do that as well?
I think they do. I don't know if it's the same company.
Yeah, I've not looked into the commercial offers of the test. Yeah. I mean, so at the moment they do the only commercially available sort of array-based profiling one is very expensive because it's a whole methylome effectively, or it's about 450,000, or the new one's 800,000 sites in the genome, and they do the whole profile, but you only need 356 or so to run the clock, at least the original one from Steve Horvath from 2013.
Okay, the company Elysium Health is the same company that makes the NAD supplement, and they do have a $500 home DNA test to tell you how old your DNA says you are. Your DNA methylation.
That's not much more than the cost of one of those arrays.
Yeah. There's one called EPIAgingUSA.com, which has one for $169. I don't know why they're that much cheaper. Maybe they're not as good.
They might be using a different assay, because you can do a targeted sequencing type 1 and run a different clock. There's a whole bunch of different versions of these clocks now. So some of them use a different technology which can be run cheaper. Yeah.
All righty. Well, we are getting close to... Oh, actually, I think we're slightly over our 2 hours. I guess we're getting close to the point where we should wrap up. So I was wondering, like, what have we not touched on that you would still like to touch on?
I think we covered a lot of ground. There's one thing that so the question that I think was raised on one of the blog posts that might have instigated the previous episode was kind of what areas might people be interested in working on in aging biology? What's an area that needs attention? And I think there's a really interesting point at the moment where a lot of descriptive work has been done in understanding how many of these mechanisms of aging work. And there's a bit of a dearth of theory, right? We've kind of gone through a period of, there was a lot of theorizing, this theory of aging, that theory of aging, and none of them were quite right. And they were all a bit, they didn't have the whole picture. But now we have a lot more descriptive information, but we don't have as much theory. So I think there's an opening for some new overarching theories of the aging process. And I suspect that these will be rooted in the field of non-equilibrium statistical mechanics. So there's an interesting book that came out quite recently from, what's the author's name? The title is Every Life is on Fire. Jeremy England, that's the name of the guy. And he works in non-equilibrium statistical mechanics and non-equilibrium thermodynamics, that kind of area. So it's understanding how like the thermodynamics of computation relates to aging. That's the area that I think is potentially very promising, because we have all of this information about the way that information processing works in biology now. We have a gap in the theory of aging, and I think it's rooted in information theory. And we have these, our biological systems are these very far from equilibrium systems that are maintained away from equilibrium by a flux of energy through them. So using ideas from non-equilibrium statistical mechanics to model aging processes is, I think, something that has a great deal of promise. But I am nowhere near physicist enough to be actually engaged in that area of research. But that's something I think has a lot of promise. So if anyone out there is skilled in the math of non-equilibrium thermodynamics and that kind of physics, the application of it to biology, I think, would be fascinating.
That sounds very promising. How would they get into that? I'm assuming there's not like an application.
I mean, to be honest, I think the starting point would be get in touch with people like Jeremy England. So there's some kind of reading around that area where, I mean, generally speaking, in academia, if you want to get into a particular area, do some reading, find out who the authors are on the papers in that area, and email them.
Okay.
A lot of scientists are working in a super niche area, and they'll respond to emails because there's not that,
Yeah. And if they have blogs, you can read them, maybe comment
exactly.
on them.
Yep. Reading the blogs and the books. So there's Jeremy England's book. There's a bunch of papers. There's a nice talk from him from MIT. That's a great starting point with a bunch of academic references in it to some of the work in this area.
Awesome. Yeah, we just talked to someone who said, they got started in Miri by emailing and asking, how can I help? And they're like, well, read all these papers for us. And they actually did it.
Yep.
And they're like, all right, yeah, let's go.
Yeah, I mean, it's often that simple, surprisingly, right? People are sort of far too afraid to just like randomly fire off an email and see what happens, because it pays off more often than you think it would.
Yeah, especially if you have.
Yeah, exactly. Yeah, it's hard to get out of that groove though. It's easy advice to give, difficult to follow. Exactly.
And just from personal experience, sometimes they just don't reply to your email, and you can't take it personally. They're busy people too.
Exactly, they're busy. They have things to do. So there's a certain trade-off between putting in enough work in the initial email to signal to that person that you are interested and competent and worth their time talking to.
Yeah, knowledgeable in the field.
Yeah, that if you convey to them that you've read some of their research and have some like potential as a collaborator that won't involve too much like additional effort from them, right? It will be a productive relationship for them to work with you. Mm-hmm. Like if you can signal that, do that, because then they'll reply to the email much more likely than if it's like, this guy's going to be a lot of work.
Yeah. Cool. All right. Well, thank you for coming on with us. I think this has been like incredibly informative. And
thank you so much for having me.
Yeah, I think this has been like the last longevity episode we did was fun and all, but the true value of that one was that we managed to get in touch with you and get you on here.
Oh, thanks. Yeah.
I tend to run my mouth a lot.
Yeah, and I'm prone to going on extended expositions, so yeah.
Mm-hmm.
Yeah. Great. Thank you. Yeah. True. Don't read my thesis. Don't subject yourself to that. I mean, feel free, but yeah.
What was the podcast again as well?
So the podcast that I'm running is Xenothesis. It's at xenothesis.com. That's spelt with an X in case anyone's unfamiliar with the spelling of Xeno. But yeah. And actually, that next week we're doing an unusual episode. It's a special one. Because Cyberpunk 2077 came out, we're doing a whole episode on cybernetics and genetic engineering modifications, and I'm going to talk a little bit about software in that context. So it's one of the things people don't talk about, like having software that you can trust running in implants that are in your body is going to be a really important thing, because the Internet of Things is a complete shit show. And when we start putting those things in our body, we're going to have a real problem. So the dystopian aspects of that, as well as the potential, is something we'll be talking about.
I would never want anything in my body hooked up to the internet.
Well, I mean, there's already Fitbits, and like the Apple Watch, I think, can track like a lot of biometrics now. And I'm wondering, since you mentioned 23andMe's privacy issues, could like insurance companies deny you based on your Fitbit info? Oh man.
Yep, that's definitely a possibility. So I mean, if you ever I don't mean to be impertinent and inviting myself back on, but if you ever want to do episodes on the subject of like software freedom and kind of how that pertains to those kind of things, the whole surveillance capitalism and the like, that area, I'll talk your ears off about that. And also academic publishing, as I managed to resist going down that rabbit hole right at the beginning. If you ever want to hear my ad hoc solutions to that problem, then let me know. Awesome. That might be a bit taxing.
Yeah.
I think that's a good place to end, though. That was you.
Yep, I did. Yes, a little bit. My little tiny contribution to the audiobook.
And thank you for that. I don't say this enough, but like the audiobook was not just my work. There was literally over 100 people that contributed voices or music or something, and it was a very large group project that I could have never pulled off without everybody's help. And
yeah, I really can't take down the opportunity to thank you for that, because aside from academic papers, I don't read much. I'm dyslexic. It takes me a little while longer to read stuff, so I listen to stuff all the time. Okay. And so without your podcast version of the Methods of Rationality, I probably wouldn't have read it and then gone down the whole rabbit hole of reading the sequences and getting into the rationality community. So thank you for that.
Well, absolutely. I'm glad I managed to have some positive effect on the world.
That's super cool.
HPML was one of few books that, like, when it got ahead of the audio version, I would actually read it because I was so interested in the storyline. But yeah, it takes a bit to get me to read something if there's not an audio version and it's not a paper.
Yeah.
Thanks. Great.
Oh, should we do a real quick thank the patron?
Who are we thanking today?
Okay. All right.
So much suspense.
If anyone hears this episode and joins the information theory of aging effort and then cures aging, this will be in part due to, well, all of our patrons, but specifically John Peterson this time. So thank you, John. You may have saved us all.
That is my fondest hope.
All right. And thanks again, Richard, and everybody for listening. And we'll see you all in 2 weeks. Bye.
Bye.
Bye, everybody.